Okay, so hi everybody. Thanks for the opportunity to be here. I'm happy. Actually, I'm mixed between anxiety and happiness. It's my first official talk outside CISA. Hope it won't mess it up. So today I'm gonna introduce a method, a Bayesian tool that we developed under supervision of my professor Guido Sanguinetti, who's the chair of the talk. And the aim was to develop an interpretable framework to analyze multi-omics data. So this is a short outline from the talk. So I'm going to speak very briefly about multi-omics data, what kind of questions it can help to answer, and what challenges it introduces when in analysis. Then I'm going to speak about the art that we use to develop our model, shared topic, which is topic modeling. And then I'm going to introduce the model. And I will focus on some main results we have, and finally draw some conclusions. So this is the central dogma that we all, the famous central dogma that we like, and we want to, like one question you can ask from this mechanism, how cells that shares the same DNA, the same information, are able to attain a high level of specificity in terms of physiology and function, what you can measure and what you can quantify in order to understand this variability. And there are a lot of measurements you can do, and in the recent 20 years, there are bulk technologies that are able to sequence a lot of these factors, ranging between epigenetics and genetics. And here in this talk, I'm mainly focused on accessible chromatin regions and RNA expression. And things became more advanced, so you have a single cell omics now, so you can do sequencing at the single cell level, so you can obtain gene expression and open chromatin regions. And then in the last 5 years, you can even obtain two modalities simultaneously in cell. Many platforms have protocols have been developed to do multi-omics sequencing, and we have this cool data now. And one question you can ask is how can you integrate such data? Because the main important thing is to understand the regulation mechanisms. So maybe this data will help us answer such questions. And as I said before, I'm going to focus on chromatin accessibility, which comes from single cell attack-seq, and the gene expression that comes from single cell RNA-seq. And briefly, this is how things work. So you have the cell, you do sequencing simultaneously, and you obtain your data. And basically, your data is per modality. Let's say for RNA, single cell RNA-seq is the expression matrix, and for open chromatin is the binary matrix. Of course, this data is not easy. It's challenging. It's highly sparse. It's especially for single cell attack-seq. It's very known. It's full of false negatives, and same thing goes for single cell RNA-seq. It's highly sparse as well. But still, we can use this data in order to study interaction, because the gene regulation mechanism remains a very important question that we want to answer in field of research. And many things has been done up till now to integrate this cool data. So first, of course, you have the deep learning approaches that uses Bayes, for example, and well, it does a very good job in reconstructing your data and clustering it, clustering cell population, but it doesn't answer the main questions, I would say, which is interaction, because it's a black box. Interpretability is not found. On the other side, you have methods that does factor analysis, like MUFA+, and this method, it kind of produce some interpretability, but it assumes a unimodal Gaussian, which is it's hard to obtain the variability across cell types from this model. So we tried to take it even further in terms of interpretability and try to account for the variability across cell population. So we actually developed shared topic, and shared topic starts from a very well class of statistical models called topic modeling that aims to annotate large numbers of electronic archives automatically. So you have your documents and you want to annotate them according to their themes. Such methods try to infer these themes starting from an abstract space, which is a topic space, and it uses a well-known algorithm, which is latent digital allocation. I'm not going to go into that. It's very famous paper cited by David Blay. But one thing I want to emphasize here is that that the data that are obtained in natural language processing is quite similar to the one we have in the multi-omics technology. So what we can do actually using topic modeling. Well, we are not the first to ask this question, of course, because people already asked this question on single-cell attack-seq data, and you have this like state-of-the-art, let's say, a method which is this topic that does analyze single-cell attack-seq using topic modeling, and it kind of offers a lot of insights on enrichment, on cell populations, and these things. But we wanted to take it more, so we moved from from topic modeling to shared topics. So we went from natural language processing to biology. So here you have very quick, your documents becomes your cells, your words becomes the regions, and the gene reads, and of course you can infer the important stuff, which is how much topic, which is still an abstract notation up to now, that we would like to interpret later. Because that's what we want, interpretability. So we want to infer how much a topic contributes to a certain cell, and how likely you can find a gene on a region contributing to certain topic. And basically, the topic is what we want. It is a biological process. So let's look even better. So this is shared topic. Shared topic, instead of one plate, as you saw in latent digital allocation, has two plates inside the main plate. One corresponds for the RNA, and one corresponds for chromatin accessibility. And in more, let's say, interpretable sense, you have your cell, you have a bunch of biological processes contributing to this cell, and the thickness of the arrows correspond to how much a biological process is actually contributing to a cell. And of course you have the gene expression data, so you can model it as coming from a Poisson distribution. So each read is modeled as generated from a Poisson distribution with an average of read of lambda. And of course, for each gene, each gene contributes differently to a different biological process. And same thing goes for regions. So regions are either found open or closed. So what's the probability of finding a region in our data? It's also depend on the biological process as well. Okay, so this is overview of what we tried to do. So here's the data, and shared topic is implemented. This graphical model I showed before using a Gibbs sampler, and then we obtain a representation in the latent space, in the topic space, for regions, genes, and cells. So what we try to do is use this cells representation to visualize cell population, and then we wanted to use the other part, the other latent space, to visualize the interactions. So this is the data set we use. We use this data coming from different single-cell multi-omics technology, from ShareSeq, 10x Genomics, and SNARE-seq. And as you can see, it's so number of regions are very high. Number of cells of order of thousands, and the number of cells range between 10,000s and thousands. So first, I'll show the part where we use single cell cell representation. So here you have the latent representation, which is which is the theta C for each cell, and you can use, of course, dimensionality reduction to visualize these things. And here you can see different UMAPs for the different 5 data sets. And okay, so the performance of cell type classification, we compared it with, let's say, 2 recently more or less developed methods, which is MUFA Plus and SCGLUE. So SCGLUE is a deep learning maze method. It uses variational autoencoders, and as we expected, the SCGLUE, and here is shared topic, and RNA only and attack only is when you account for a model which only is based on RNA, like the left part of our model, or attack only, which is the right part of our model. And same thing goes for SCGLUE. If you only take one modality, not the joint. And as expected, the performance of a variational autoencoder will most of the time be the best one in classifying. So this is the result of K and classifier. And but one thing to notice is that in many data sets we are not even with our model that is not based on a deep learning method, we are not very far from reaching the performance of SCGLUE. That's the first thing. And one thing is that shared topic is able to outperform with 2 modalities the other if you take the modality separated, which is also a good thing. And then the last part, which is using the regions and the genes representation to reconstruct the regulatory mechanisms. So what we did is basically we use the latent presentation to associate the genes and regions to a biological process, and we then obtain a joint distribution that tells us how much a region is contributing to a certain gene that belongs to the same topic. So first thing, we want to associate genes and region to a biological process. So what we try to is to compute the entropy of each gene and region across the topics. So we expect that entropy that have the genes and regions that have the lowest entropies are not uniformly distributed across topic. Like this example, so you have a gene here, and you have two lambdas that are clearly relatively higher than the other lambdas. So you can say, okay, we have 2 topics here, which are basically biological processes that this gene is contributing to. So basically, this gene has a very low entropy, and basically you can. And so the green line here shows you the case of uniform distribution across topics, and you can see all the genes are far from being uniform, but some of them have very strong membership to a biological process. So we took the top 30% genes that have the lowest entropy and say, okay, these genes have clearly contributing to a certain biological process. And of course, and after assigning, so one example I'm going to show here is coming from the B lymphoma data set, topic 13, and so to annotate this topic, to give some interpretability to this topic, you look at the gene set that is associated with this topic, and you can carry gene enrichment analysis. And as you can see, there's 2 biological processes that we are interested in to see here, where is production of cytokine and immune response to regulating signal pathway, which kind of show that there's some sort of inflammation happening. And of course, after and then we look at the genes and we look at the genes and regions that are allocated in the same topic, and then we look at the probability of the region to be open in that topic because it has a stronger membership in it. So you have this the probability of it is 5 star, which is the highest probability, and then the other 2 here we're focusing on promoters. So the other two promoters have have lower contribution to this FOXP1 gene. And one good observation that we did as well, we looked at the UMAP and we looked at the theta 13 for all cells, and we found them enriched in the tumor B cells types. So if I go back to here, so this is the tumor B cells, and we found that topic 31 is enriched in the cells, and yeah. So conclusions: shared topic is able to provide a very, let's say, a methodology to get some interpretation, and develop, let's say, it's a method developed to find regulatory mechanisms in the cell using multi-omics data. And we hope that we will be able to integrate even more data inside shared topics like DNA methylation and these things. And yeah, so with the view from our office, I finish. Questions?
The same sea that you see. Thank you very much, Nour, and well done in making it through. Let's see if you make it through the questions now.
Thanks, boss.
So, any questions for Nour? Because I'm kind of in a situation where I can't ask a question.
Could you go over the topic modeling again? I was trying to understand the, I didn't quite, this is just because I don't understand it very well, but how does it work? The words are the genes?
Yes, can I? Okay, this one here. Yes, so yeah, the words and the regions, the open chromatin regions, and their gene reads are basically the words in the document.
And then is it like, it might be off topic, but is it
It's okay.
like a bag of words?
Yes, exactly.
Okay.
Yes, exactly.
Yeah, it's as if you have a document in two languages, essentially.
Yeah. So does context matter? So one of the things that I think,
Okay.
I don't know for sure, but I thought bag of words had trouble when context matters a lot, like when the same word.
Yeah, there are some times where LDA doesn't, even topic modeling in general doesn't do very great, but well, it still does some in certain settings also a very good job.
Okay.
So ChatGPT is not based on topic.
That's for sure.
It's not a good generator of text. It's
Yeah.
a good annotator.
Yes,
It's
exactly.
good for interpreting text.
Okay.
More curious on the part where your example of FOXP.
Yes, of course.
Where you have the contribution from different promoters to that. I wonder, I guess maybe I missed that part, like where did the probability of different promoters contribute to the gene?
Yes,
stuff?
yes,
Is
yes,
there
yes.
weight for probability?
Yes, so you have you have FOXP1, and these promoters are assigned to a topic, which is, so they have a clear membership. They are contributing to topic 13. It's an abstract notation until we do some annotation, right? And then you say, okay, I have these promoters that are around gene FOXP1, and I look at their probabilities of being open in that topic, and then I find that there's one that clearly is better than the others to be found to be contributing to FOXP1 in this biological process.
And because we're restricting to nearby promoters, you also, but it essentially also can be like larger.
You can also take you can take larger circles of course, and then you might find more promoters, but then you will find that fire is kind of dropping a bit down if you go there, if you go far, especially regarding the biggest regarding. So you might find phi's that are higher than these, but not higher than this one. Okay. Because I know that FOXP1 has a lot of I think I've seen a lot of promoters associated to FOXP1.
In fact, it's kind of the usage of promoters in FOXP1 and in is something that
And to
some kind of heuristic for the, let's say, the number of topics that you're looking
Yes,
for?
yes. So we use WIC, which is it's a way to compute the likelihood. It's a model selection method. So you compute the likelihood and you penalize by the number of the complexity of the model.
It's not really a heuristic. It's
Yeah, that's not heuristic.
but we had a way to
WIC.
What is it? Watanabe? Widely applicable information criteria or Watanabe information. They always do this trick.
No, because you want to add topic that get assigned with low entropy, right? Why don't you put entropy also in the like in the ICL platform?
I mean,
Topic assignment with so that's a gene assignment to a topic.
Right.
So you hope that genes are not, so uninteresting genes will be, okay, I'm not expressed anywhere, so zero. Interesting genes will be, I'm expressed in one topic. Gabriele?
I might have missed something, but are you also using, or could you use the sequence motives in the region itself, or are you just using the region?
no, I'm not using the sequence. I mean, you mean annotating the?
Like a binding motif.
No, we're not using. No, we just annotate the regions and that's it. But no motif.
We could look at the sequence of the preferred check. Right, so we actually have
We
a
have the-
speaker.
we have the
And it's a very exciting conference. You never know what's coming.
Okay. Thanks, Julio.